So now we are entering, let's say, some more technical discussions, scientific discussions. So we'll have two presentations. The first one by Sven Borgos from SINTEF, Norway, and then the second one by Neil. So Sven, the floor is yours. 10 minutes.
Thank you, Patrick, and thank you everyone for coming. As Patrick mentioned, we're going to split this session in two. So first I'm going to tell you a little bit about physical and chemical characterization, and that's also going to be a composite because the slides on physical characterization were provided by Fanny sitting at the front here. And then afterwards, Neil is going to go to the in vitro and in vivo side of things, and we're going to try to tie it a bit together. So we decided to look at a case study of liposomal irinotecan. And as I'm sure most of you are aware, there are a range of physical and chemical aspects of nanomedicines that are defining and that are interesting to look into. There are the purely intrinsic physical properties like size and shape. There's surface modifications. And then there's the chemical composition. And then when the particles start interacting, for instance, with a more complex biological system, it also starts exchanging components, drugs typically. So it's really there's a structured chemistry in nanoparticle, and that makes it quite a bit more complicated than a dissolved small molecule drug. These factors are biologically relevant. Just think about how nature uses shape in order to obtain specific effects, and how many shapes are actually available in natural forms. And it is also well known that both size and shape can dramatically affect things like biodistribution. So on a very, very high level readout, the size and the shape will actually have an effect. So the objectives of physical characterization would typically be these in the first round at least. For the pristine nanoparticles, you want to look at batch-to-batch consistency. You want to look at things like particle size and particle size distribution, because hardly any nanoparticles are completely monodisperse. You want to look at shape. You want to look at surface charge. And you want to look at whether the coating is actually coming off or not. These are kind of fundamental things. Then you also want to look at how they start behaving in presence of, for instance, plasma proteins, whether they start aggregating, which could be kind of critical in the body if you have large aggregates of nanoparticles rather than distributed like they should be. Now, there are some challenges when it comes to physical characterization. As I mentioned, we're not only looking at size, but also things like size distribution and shape. And for multimodal samples, this begins to be challenging. If you have complex distribution, you want to start resolving them. And that could be discriminating based on shape, because effectively you could have particles of the same volume but with different shapes, and they could have different effects. You want to start looking at particle size distribution differences in the presence of serum proteins. When you have adherence of proteins to a surface, the effective particle size goes up, for instance. It could be that simple. And also, for small nanoparticles, you start to get interference from the large proteins themselves when it comes to size measurements. Also, a lot of measurements techniques assume spherical particles. A lot of the sizing techniques assume that they are spherically symmetrical. So not all particles are spherical, and how then do you adapt for that? And how do you actually define size for a non-spherical particle, for an elongated, for a cylinder, for instance? What is the most relevant measure of size? So now this is liposomal doxorubicin. You have batch mode DLS, which is by far the most common way of measuring size. You have a nanoparticle tracking analysis, which is kind of coming up. And then you have a separation-based technique called field flow fractionation, which actually separates your population upstream of online detectors. Then you can also look at it by cryo-TEM, so actually get electron pictures of them, and you can start making detailed distribution like this. So this formulation has a reasonably nice uniform size distribution. Now, when you start to look at the Irinotecan liposomal formulation, we do batch mode DLS, and it all looks very nice. The polydispersity is quite low. You have a nice distribution here. Now, if you stop here, you will actually miss something. We decided to look a bit further into this, and already in the NTA plot, you can see that this is not really one peak anymore. There is something happening up here. Then we subject it to FFF, so separation-based detection with the multi-angle light scattering, and you can very clearly see that there's actually two populations in here, even though DLS makes it look like there's one homogeneous population. So this warrants to really dig a bit more into detail and do electron microscopy, and here it becomes really apparent that this is actually a mixture of two populations where one is spherical and one is more coffee bean shaped. And please note that you would not be able to observe this in batch mode DLS. So this is the aspect ratio. There's a cutoff here of 1.1. So we define everything with the aspect ratio between the long and the short axis as spherical. And you can see that there is a significant part of the population which has the coffee bean shape. So I'm not going into all the details, but just to let you know that DLS would only detect one peak. Other complementary techniques will give you more information, and it really shows the value of adding complementary orthogonal techniques. When it comes to chemical characterization, you want to look at things like drug loading, free versus bound drug, particle composition, obviously, and in the physiological context, you want to look at drug release, which was already mentioned by Susanna as one important parameter to look at as identified by the regulators. Very briefly on Irinotecan. Irinotecan is a cytotoxic drug, but it's not really the drug. It's a prodrug of a compound called SN38, and the cytotoxic activity of SN38 is about 2,000 times higher than Irinotecan. So they added this group here in order to increase solubility and increase pharmacokinetics of the drug. But this is the active compound. When body wants to get rid of it, it puts on it's glucuronidated, and this increases obviously the solubility a lot, and it's excreted. And a tricky bit about Irinotecan is that the glucuronidated compound is excreted into the intestines, and in the intestine you have bacteria which chops off the sugar moiety, regenerates SN38, and gives you diarrhea. So it's a bit of a tricky compound to handle. Not going into the details about this, we used LCMSMS with isotope label internal standards and got to total drug concentration of 4.33 milligrams per milliliter, which was really close to the specifications. So that's okay. Then there's the free bound drug, which I mentioned that we want to do as a standard. So using ultrafiltration in centrifugation-based spin-off filters, cutoff filters. And again, not going into the details, but we can measure that a little bit less than 1% of the drug exists free, so not encapsulated in this formulation. Now, I want to spend a little bit of time on this one. We are not going to go through the whole data set, but what we did was we took cell culture, we exposed it to either the encapsulated irinotecan, the free irinotecan, or even the metabolite, the most active metabolite. Then we let the cells grow for a while, and we measured the actual concentrations of the irinotecan, the SN38, and the glucuronide in the aqueous phase, and we also tried to do it in the extracted cell layer. Now, these are consider them semi-quantitative between the three types of samples. They are quantitative in a relative fashion, but consider them, you will not basically be able to close the mass balance here. But it does give you an interesting indication of the fact that the cells are metabolizing and re-excreting compounds. So the take-home message is that the cells are not experiencing what you put into your cell culture. They are converting your compound. And obviously, when you want to look at cell killing and you know that the metabolite SN38 is a lot more cytotoxic than what you added, this becomes important to understand the cell killing, for instance, to understand the cytotoxicity. And on the in vivo side, this translates into pharmacokinetics, which is a really well-established field. So we have identified it as a general objective for EUNCL to try to add more mechanistic understanding here. Not only pour the drug into your sample and measure whether it dies or not, but actually try to understand why do we see the effects that we do. So integration of physicochemical assays with the cell-based and in vivo assays is key to us. And again, we have to emphasize characterization in the most relevant context is important. So not only pristine nanoparticles in PBS. There is further work going on about this. These are the things that you should take away from this talk. Orthogonal assays are needed for multimodal samples. Do not trust DLS by itself. Our cascade includes several of these orthogonal assays. EUNCL promotes the use of SOPs, as has been mentioned several times already. And these are available online on the website. So also, measurements should be done in the most relevant context. So in the presence of plasma proteins, if you have things that have strong protein binding. And again, integration of assays will give you a lot more relevant information than just doing them blindly separately. So yeah, thanks.